Once again, class 12 video. The next topic is the MEGASPOROGENESIS or development of MEGASPOROGENESIS or development of MEGASPOROGENESIS. Okay, in my previous class, I already told you about. In the previous class, we are discussing about this embryo structure, anatomic overview. All this structure is clear here. This is a skeletal end, very important micropylori. When we draw the diagram, it must be shown. You have to show both the ends separately in the diagram. That is compulsory. Okay, you have to mention the skeletal end and the micropylori. So this is the one cell present in the nucleus. Nucleus is the part which provides the nutrients which further develop to form embryo. This is the one important cell enlarged in the size which is closer to micropylori end. Closer to micropylori end, it means it has ability to develop as a micro. Little bit right about the text of this. The process of what is the megasporogenesis? The process of formation of megaspore from megaspore mother cell. What is megasporogenesis? It is the process. It is the process of formation of megaspore. Megaspore. What is megaspore? This is actually female gamete. Megaspore from megaspore mother cell. Megaspore mother cell. Mother cell. That is MMC. It's called. That is microsporogenesis. Simple definition is here. In ovule, the single microspore mother cell, which is closer to the micropylori end region, it gets enlarged. Look at here. This is the single cell which gets enlarged. So what happened in the ovule? The cell, which is all the cells which are present in the ovule, it has potential to form the megaspore. Only a single megaspore mother cell MMC present near the micropylori end. This is important. Near the micropylori end. What is micropylori end? It is an opening. Okay, it is an opening. So one cell which is present near the micropylori end, it gets enlarged. In ovule, a single microspore mother cell present near the micropylori end gets enlarged. Okay, look at here. This is the blue cell. It gets enlarged. Why it gets enlarged? Because it has to form the megaspore. That is a female one. Then what happens? This cell containing dense cytoplasm and the prominent nucleus. What is present in this cell? This cell contains dense cytoplasm and a prominent nucleus. A prominent nucleus. Okay, the prominent nucleus is present here. Prominent nucleus. So this cell contains the dense cytoplasm and the prominent nucleus. So what happened here? Here the meiotic division goes on. You know very well what happened after the first meiotic. One cell is divided into two. In the microsporogenesis, we call it the dyad. So here also one cell divided into two to form dyad. Two cells are formed. Then again the second meiotic is going on And after that, after second meiotic, 4 cells are formed, and they are called the tetrad. Okay, so due to this division, which is called the meiotic division, the first meiotic and second meiotic, tetrad is formed. What happened next? Due to meiotic division, after first meiotic, dyad is formed. Dyad is formed, means one cell is divided into 2, and then after second meiotic, tetrad is formed. Okay. Now one cell divided into 2 and after first meiotic, then two cell divided into 4 after the second meiotic. What about the tetrad? The tetrads are always linear in position. Tetrads are always linear. Linear means they are in the straight line. What is the position of tetrad? It is linear in position. It is always in the straight line. Okay. Now what happened next? Out of the 4 cells, in majority of the flowering plants, only one cell is considered as a megaspore because only one cell is functional or it is survival. Rest of the 3 cells get degenerated. So what happened? In majority of flowering plant, only one cell out of all these 4 cells, only one cell is functional or survival to form megaspore, the female plant. Okay, then what happened to the rest of the 3 cells? The rest of the 3 cells get degenerate. Okay, what happens? one cell which is present in the nucleus, one cell which is present acts as a megaspore mother cell. It gets enlarged, contain dense cytoplasm and prominent nucleus, and meiotic division goes on in that cell. Due to post meiotic, one cell is differentiated into the dyad, then 2 cells differentiated in the tetrad. So 4 cells are formed, in which 4 cells all are the linear manner, and in the 4 cells, only one cell has capability to form megaspore. One cell is functional, one cell is active, survival, and rest of the 3 cells get degenerate. Okay, then only one cell remains here to form the embryo. It means only one megaspore develops from embryo. So it is called the monosporic development. Okay, only one. I write it here. Only one cell is developed here. So this development is called monosporic development. Okay, this is all about the detailing about the development of the megaspore, how the megaspore is formed. Now let's move towards the diagram. Okay, now diagrammatic explanation. What is going on in the process? What is going on? This is the ovule. Outside this ovule, all the cells that we call the nucleus. We call the nucleus. This end is called the micropylar end. Must remember, you have to mention in each diagram which end is indicate which part. This is the callosal end. Okay, this is the ovule. One of the cells which is present here in the ovule near the micropylar end gets enlarged. Okay, one of the cells gets enlarged, having prominent nucleus and dense cytoplasm, and this is called megaspore mother cell. This megaspore mother cell further moves to the division called the meiotic 1 and then meiotic 2. What happened next? This is divided into 2. All the nucleus is here. This whole nucleus is outside this. Okay, and it is divided into 2. This is from one cell, 2 cells formed. This is because of the meiotic 1. Meiotic 1. And we call it megaspore. Megaspore diet. Must remember, this is a micropylar end and this is the chelical end that you have to mention. Then what happens? Again, the one division is going on, that is called the meiotic 2. Meiotic 2. As a result of this meiotic 2, the 4 cells are formed. 4 cells. Nucleus is present again. The outside of this, the nucleus is here. Okay. This all are the nucleus. And these are called the diet because it is 2 in number, and this is called the tetrad because it is 4 in number. 1, 2, 3, 4. This is called the tetrad or megaspore. Megaspore tetrad. Megaspore tetrad. Nucleus is already there. And what happened now? Out of the 4 cells, only 1 cell has the ability to survive or to activate. The rest of the cells get degenerated. What happened next? After the formation of megaspore tetrad, just 4 out of 4 cells, 1, 2, 3, 4, I need to separate here. 1, 2, 3 and 4. Okay. The 4 tetrads are there. Out of this 4, 3 cells degenerate. And only 1 cell gets dead. Only 1 cell gets dead. 3 cells get degenerate and only 1 cell is able to survive. Only 1 cell. The cell which is in the micropylar end is able to survive. Now what happens? The surviving cell, some changes occur in this surviving cell. This is the one single surviving cell having 1 nucleus. Okay. Now what happens? 3 types of the division is going on. 3 divisions and 3 types of the karyokinesis is there. Karyokinesis. What is karyokinesis? Karyokinesis means division in nucleus. Okay. What happens? This 1 nucleus is divided into 2. After first division, then this 2 nucleus divided into 4 after second karyokinesis, and after the third karyokinesis. After the third karyokinesis, this 2 is divided to form 4. This 2 form 4 nucleus. This 2 is also formed 4 nucleus. So what happened here? The 3 karyokinesis division takes place. Karyokinesis means that is the division in the nucleus. So what happens? The 3 cells get degenerated and only 1 cell remains here, which is active, and that active cell, this is one active cell is here. In active cell, nucleus is present, cytoplasm is present, no cytoplasmic division takes place at that time. As no cytoplasmic division, then what happens? The karyokinesis is there. This is the process of karyokinesis. And what is karyokinesis? As I told you, Karyokinesis means the division in nucleus. And due to division, three karyokinesis takes place. And because of the three karyokinesis, the one nucleus is divided into two after first karyokinesis. Two nucleus is divided into four after second karyokinesis. And then third karyokinesis, four nucleus is divided into eight. So what happens? So one cell is there having the 8 nucleus in it. Okay. After this, the process which is going on is called cytokinesis. What is cytokinesis? Cytokinesis is the division in cytoplasm. What happens to next? This is a simple cell. I'm going to rub little bit there to draw the diagram. Okay. Here we have one cell. Okay, we have this one cell having 8 nucleus. Okay, what happened? The 3 nucleus move to this end. 1, 2, and 3. The 3 nucleuses are moved to one end. One end, that end is called the chelazial end. Okay, rest of the 3 nucleus that move towards this area, other end, and 2 remain here only. Total 8 nucleuses are there. 3 nucleuses move towards the one end, 3 nucleus move towards the another end, and 2 nucleus remain in the center. The 3 nucleuses that move towards the chelazial end, they are called antipodal. Antipodal cell. Okay. The 3 nucleus which moves towards the micropylar end, this end is called the micropylar end. 3 nucleuses that move towards the micropylar end, they are called the egg apparatus. They are made up of 3 compositions. 2 synergids are there. 2 synergids are there, and 1 is the egg cell. And combinedly these are called egg apparatus. Okay, out of the 8 nucleuses are there, and in 8 nucleuses, 3 move towards the chelazial end, and that is called the antipodal cell. 3 moves towards the micropylar end, which is composed of synergids and egg cells, and they are called egg apparatus. And 2 remain in the center, and that is called the polar nuclei. Polar nuclei. Okay, this is the complete development of the embryo. Now, what is the structure of embryo after this development? The structure of embryo after this development. An important diagram from this topic, that is the structure of embryo. Okay, look at this picture carefully. We have to draw it again in proper manner to show the embryo structure. This is the outermost membrane which is present. What happened here? This end is from the antipodal cell, and this end is from the chelazial. This is micropylar end. This is the chelazial end, and this is the micropylar end. Okay, 3 cells are here. Look beautifully. Because the decoration must be there in the diagram for the first view. When we draw it, these are the 3 antipodal cells. 1, 2, and this 3. The 3 antipodal cells there. This is the outer membrane. This end is called the microphylar end. 2 nuclei is present in the center. 2 nuclei which is present in the center. And 3 nucleus move towards the microphylar end. This is called the egg cell. Egg cell. This is called synergid. This is polar nuclei. Polar nuclei. This is antipodal cell. This is the complete structure of an embryo. Now look at here carefully. What is an embryo? It is a complete developed embryo is actually 8 nucleated and 7 cell structure. 8 nucleated, 1, 2, 3, 4, 5, 6, 7 and 8. 8 nucleated and with 8 nucleus number of cells is 7. 1, 2, 3, 4, 5, 6 and 1 whole. This is the 7. It is called the 8 nucleated 7 cell structure. Let's talk about this, about the final structure of the embryo. What is the final structure of the embryo? It is 8 nucleated and 7 cell structure. The final structure of this. This is the synergid. Synergid play an important role at the time of the formation of folate tube. So what is the structure of the embryo sac or this is the embryo sac or female gametophyte. Female gametophyte. Okay, this embryo sac. So what is the peculiarity about this embryo sac? What happens after first meiotic division is going on, due to which the dyad is formed. After second meiotic, tetrad is formed. What happened then? Out of tetrad, 4 cells, 3 cells get degenerated. Only 1 cell is developed for the surviving. What happens for the surviving cell having the karyokinesis? Okay. What happened next? The 1 cell, surviving cell or functional cell, surviving after tetrad. Continuous karyokinesis up to 3 in number to form 8 nucleus. Okay, to form 8 nucleus. That is explained. Okay, 8 nucleus is formed. It is 8 nucleus. Out of this 8 nucleus, 3 nucleus move towards chalazial end called antipodal. 3 nucleus move towards microphylar end called synergid and egg cell. And 2 nucleus to nucleus remain in center called polar body, polar nucleus. Okay, polar nucleus. So this is the condition. Condition after the division going on here. This area, this is called the filiform apparatus. This is called the filiform apparatus. Why this is called the filiform apparatus? Because it is active. Okay, it is active at the time of the pollen tube formation. This is the place which allows the pollen tube to get inserted inside the female nucleus. Okay, it is present in micropylar end and it is a little bit thickened portion. It is a little bit swollen portion and this is called the filiform apparatus. Here, filiform apparatus is present. Filiform apparatus is present. Okay, before writing this about the filiform apparatus, must write the condition of the embryo. This embryo is called 8 nucleated 7-cell structure. 8 nucleated and 7-cell structure. Why it is called the 8 nucleated and 7-cell structure? Because total 8 nucleus are present here and number of cells is called the number of cells present is called the 7. It is a polygonium type of embryo. It is polygonium type of embryo. I already told you it is monosporic. Monosporic means only one cell gets developed to form from megaspore mother cell. Only one cell is developed to form one embryo. So one filiform apparatus, this is spatial cells are present here. This filiform apparatus, this is a filiform apparatus. And what is the role of this filiform apparatus? It helps the pollen tube to get inserted in the female nucleus. So we must write one line here also. The more important thing you have to write here, filiform apparatus is present. What is the filiform apparatus? It is a swollen part. Near this X cell, the two cells are two nucleus are there. It gets swollen. And it's swollen. Why it gets swollen? Because it increases its thickness. Because it allows the pollen to get inserted into it. A filiform apparatus is present, which is active or present through which pollen tube inserted in the female. So what is megasporogenesis? Megasporogenesis is the process of formation of the megaspore. What happens in this megaspore mother cell in the nucleus? Only one cell which is getting enlarged is near the micropylar end. That is called the megaspore mother cell. First meiotic division is going on. 2 cells are formed. Second meiotic division is going on. 4 cells are formed. After the 4 cells, it gets degenerated. Only one remains active. It gets enlarged. Then 3 karyokinesis is going on. The enlarged is the nucleus is haploid cell. Must remember, this is enlarged is the haploid. All the cells are the haploid. Okay, because it is formed as a result of the meiotic division. Okay, all the cells, whether it is a microspore or the megaspore, they are the haploid because it is formed as a result of the meiotic division. Okay, so this is haploid cell. 3 karyokinesis is going on simultaneously. 1 nucleus divided into 2, 2 nucleus divided into 4, and then the 4 nucleus divided into 8. At the end of this process, we are having the 8 nucleus and 1 single cell. Then what happens? 3 of the nucleus move towards the chelaisal end to form antibodies. 3 of the nucleus move in the micropylar end to form the X cell and the synergid, and 2 cells remain there only. Once again, I repeat it. This structure is called the 8 nucleated 7-cell structure because 8 nucleus is present. 1, 2, 3, 4, 5, 6, 7 and 8. But only 7 cells are present. 1, 2, 3, 4, 5, 6, and whole is the 7. Only 7 cells are present. This condition is called the 8 nucleated 7 cell structure. Must remember, this is a monosporic development. It is also called the polygonium embryo. Okay, all for today. Bye bye.